The ReproTox knowledge graph is the result of a partnership between 4 project groups within the Common Fund Data Ecosystem, CFDE. The 4 projects are the Library of Integrated Network-Based Cellular Signatures, LINKS, Illuminating the Druggable Genome, IDG, Kids First, and Stimulating Peripheral Activity to Relieve Conditions, SPARC. This video provides a brief outline of this knowledge graph and is a key step ahead of collecting feedback from the community about its strengths and future potential. The ReproTox graph links knowledge about birth defects with drugs and genes that are likely to be implicated in their teratogenicity. The workflow that generated this graph is explained in the 2023 Communications Medicine publication featured here and linked below. In practice, therefore, this video will act as a quick guide to this paper, describing the 6 core datasets that were brought together to generate over 500 cliques, network triads of closer associated birth defects, drugs, and genes. And before we take a closer look at these 6 datasets, it is important to note the pervasive role in ReproTox that the links derived small molecule similarity scores on the basis of the pairwise resemblance of gene expression perturbation signatures have played in augmenting the prediction of functional clusters of small molecules beyond the well-documented limitations of conventional structural similarity calculations. So let us briefly highlight the 6 core datasets that underpin the ReproTox calculations. The first, labeled A here, is a list of birth defect-related phenotypes and associated relative frequencies, curated from the Kids First Pediatric Research Program cohort. The mapping of these birth defect terms onto the Human Phenotype Ontology, HPO, marks a key step in standardizing representation, accessing birth defect mentions in the literature, and retrieving gene lists associated with these phenotypes by reputable external sources that annotate with the HPO. The second dataset was curated from various sources including DrugCentral, drugs.com, DrugShot, FAIRS, and the National Birth Defects Registry to build a master list of teratogens and xenobiotics with potential to cause birth defects. Furthermore, data from PubMed co-mentions were analyzed to ensure robust evidence of drug birth defect associations. For the third set, human phenotype to gene associations were retrieved from multiple sources including Pharos, OMIM, Orphanet, ClinVar, Diseases, Decipher, the American Heart Association, and GeneShot, to leverage curated links between HPO terms and human genes. The fourth set built a mapping between small molecules of defined structure and their known targets, as represented in the TCRD database, but steering clear of multi-component ion channels or transporters. The fifth set takes into account the notion of gene intolerance, that is, the likelihood that any structural functional interference to a gene gives rise to a pathological outcome. By comparing the observed number of variants in each gene to the expected number based on statistical models, it is possible to assign scores on the deviation of observed variants from the expected number. This score helps summarize the gene's total variation and the overall impact of this variation on gene function, on the basis that the more variants a gene has, the more tolerant are its products to perturbation. The sixth and final set brings into focus an essential feature that most teratogenic factors share, the crossing of the placental barrier. Obtaining experimentally determined evidence that a molecule shows significant placental crossing is of course challenging, so such reference datasets would be relatively small. In this step, therefore, a semi-supervised learning approach was used to predict placental crossing for a large set of molecules in the links resource, on the basis of similarity based on gene co-expression and chemical structure, starting from a reference set of 143 small molecules that are known to cross the placenta. The ReproTox knowledge graph of birth defects, drugs, and genes was built from these 6 datasets, capturing associations between genes and birth defects based on mutations that are known to cause that phenotype. In this chart, lined blue nodes represent birth defect terms, orange nodes, genes, and pink nodes, small molecules. Red lines with diamond heads indicate that a small molecule upregulates the gene, and plungers indicate downregulation. Grey arrows link birth defects to small molecules or genes whose pathological variants induce teratogenic outcomes. And finally, resources generated by the ReproTox effort include the scripts running the relevant calculations and data transforms, downloadable via GitHub here, the resulting component knowledge graphs available via the ReproTox website, and a graphical user interface that allows for the interactive browsing, querying, and downloading of subnetworks of interest from the graph. So, that was a brief outline of the ReproTox resource and associated publication, as a first step to community feedback and market data gathering by the Common Fund data ecosystem.